GeoVax recently published an article in a recognized journal regarding the efficacy of a multi-antigen vaccine. What were the takeaways from this study?
Takeaways from the study were that a multi-antigen approach induces not only the antibody side of the immune system, but also the cellular side, or T cell, which is what is critically important. Our product that is currently in the clinic, so CM04S1 as we refer to it, that utilizes two antigens, the spike antigen and the nucleocapsid, and that gives a very broad response as well as longer lasting. But what we've also had in development, at this point still in preclinical, but it was involved in that vaccines article very highly, is one that is a 3 antigen. That includes the spike protein, the membrane, and the envelope antigen. And that vaccine is at least demonstrating in a preclinical modality that it could be the first step towards a pan or universal coronavirus vaccine. So we're quite excited about our portfolio of multi-antigen vaccines against SARS-CoV-2 and other coronaviruses in that instance.
And you have a clinical trial that is going on or soon to start for immunocompromised patients in this area. What's the plan for that, and what should we expect in terms of milestones?
Yeah, we actually have 2 phase 2 trials currently underway, actively underway among immunocompromised patients. One is blood cancer, hematologic cancer patients, and in that one they're being prepped to receive stem cell transplantation or CAR-T therapy. When you do that with patients, you deplete their immune system or their antibody immune system. So these individuals are put into a state where they have no protection derived from receiving, for instance, mRNA vaccine. So in this one, we're doing a direct randomized comparison of our vaccine, because it induces a very strong T cell response, also against either the Pfizer or Moderna vaccine. And we're looking at that as an alternative because that will provide protection to these patients they otherwise will not have. The second immunocompromised trial is among chronic lymphocytic leukemia patients. That is a well-recognized blood cancer, a group of patients who have a very debilitating situation resulting from their disease and even the therapy that they receive. And in that one, it's a randomized comparison against the Pfizer vaccine. We recently announced and released the interim results. In fact, they were presented just recently at the World Vaccine Congress, which was in Washington, D.C. And in that study, what we saw happen was the Data Safety Monitoring Board recommended and subsequently halted the Pfizer arm because it was ineffective, and ours had exceeded the targeted endpoint, and the remainder of the study is simply utilizing our vaccine. We only need about 24 patients more to complete that trial. We're hoping that will occur yet this year, and we look forward to those final results, because if they if they replicate what we learn in the interim results, we anticipate going forward with a registration-oriented trial with CLL patients. So we're quite excited about our vaccine performing what we're seeing now with clinical data in immunocompromised populations.
Okay. You know, just another thing on the article that you guys contributed to. I think they identified 4 different antigens. I think there was the envelope, the nucleocapsid, and the 2 others that you had already previously.
The
membrane and the spike, right? Yeah. Is there any? Do you think in the future that all 4 of those might be appropriate, or is it just better to use 3? I guess you probably optimized it for what works best, right?
I would say, first of all, we don't know. Adding additional antigens into a construct of a vaccine, and not all vaccine platforms can be multi-antigen. mRNA can only accept 1 antigen at a time, and other technologies are.
Which may be why you outdid Pfizer, right?
Exactly. And so, well, we did that because they were the authorized, so that's what's most important. But most vaccines typically focus on inducing an antibody response. But there are various infectious threats or conditions that you need a broader sort of immune response for the patients, for the benefit of patients. And that means you've got to utilize, and you need to utilize, cellular immunity. So that becomes very important on that. There's a challenge technically in constructing a vaccine that every time, at least with MVA, which is what we're utilizing, when you add another antigen, you've got to also ensure you have a proper balance of those antigens, because you don't want them biologically to start sort of canceling out what one might do versus the other. We've done development vaccines before up to include up to 5 different antigens in a construct against a particular infectious disease. So more than 2 can be done, more than 3 can be done. We believe you can do up to 8 or 10, but it's a very tricky type of aspect to do. I would say, I think this is where artificial intelligence might come in to be helpful.
Right, to optimize
because
the
our
mix
team,
of antigens.
Our
Yeah.
team is trying to utilize that right now. So we're trying to take advantage of AI, which most of us don't even know what it really means, but we figure that's the type of tool to be able to, using big data and the capability of AI, to be able to look at what all is known out there, and can we more effectively pull all that information together and understand an improved manner and faster manner of being able to utilize multiple antigens into an MVA construct and do that faster, and let's say also do that more efficiently. And that becomes very important. So time will tell. We'll keep you updated on this, but we're trying to utilize AI to better understand that, whereas historically it's been literally someone who's a virologist working and doing the experiments and utilizing what they know in their head. So we're trying to bring all this other stuff together also to speed up the process, make it more efficient, and be able to do, let's say, improved vaccines that are multi-antigen.